Welcome to Clinical Trialblazers, a podcast hosted by Biotech, where we dive deep into the journeys of visionaries shaping clinical research and biotech innovation. I am your host, Alberto Grignolo. Today, I'm excited to be joined by Dr. Ross Breckenridge, CEO of Arjuna Therapeutics. Ross is a physician, cardiac biologist, and biotech executive who trained in medicine at Oxford University in the UK and was later awarded a PhD at Cambridge University, and whose journey spans academic medicine, translational research, and therapeutic innovation. For nearly 2 decades, Ross held academic and clinical positions at the UK National Institute for Medical Research and went on to train as a clinical pharmacologist at University College London. He then moved into biotech leadership, serving as Chief Medical Officer and CEO at Silver Creek Pharmaceuticals, where he advanced regenerative therapies for acute and chronic diseases. Now, as CEO of Arjuna Therapeutics, Ross is pioneering a novel approach to precision oncology, therapeutic molecular clusters. His path reflects a deep commitment to both science and patients, and he joins us to share the story behind Arjuna and the bold ideas driving it forward. Ross, thank you for joining us.
Thank you. Pleasure to be here.
Let's get started, and let's start with your career path, if we could. You moved from academic cardiac biology into oncology biotech. Was that shift intentional, or did it emerge through maybe unexpected opportunities?
Well, I guess it's the same as everybody. There's a sort of a pathway with opportunities offered that you decline or you take. And the way that medical training is organized in the UK, you're kind of offered a smorgasbord of potential specialties very early on in your career, and you really get to choose your direction. But I always knew that I was very interested in basic science as well as clinical medicine. So in the end, biotech, if you actually want to make an actual difference, biotech is the obvious place. How I got there, I mean, it's a random walk, I guess.
Were there any mentors or role models who particularly inspired you in the early stages and over time? I might note you have a famous name, and many listeners will wonder if you have any relation to Sir Alberto Breckenridge.
Yeah, well, I'm my father, so yes, a very profound relationship. And my surname was actually rather difficult to hide from as a medic, because everybody had heard of him, and a lot of senior people had worked with him. Thankfully, he was somebody that everybody enjoyed working with. That actually worked out very well for me. Certainly, it is a double-edged sword having a famous parent in medicine, but I'm very, very lucky the father that I had. I'm also very lucky that he transferred his childlike love of biology to me very early on, and I still have that.
Let's move to some insights that you might have picked up along the way, perhaps as a result of your long tenures at the National Institute for Medical Research and UCL. What key insights from that work have carried over into your current focus on cancer?
The work that I was doing at NIMR was really looking at molecular adaptation of cardiomyocytes to hypoxia and also to metabolic challenges. And really, that's pretty much what our scientific focus is at Arjuna, except with cancer molecules. The mitochondria are the same. The metabolic pathways are the same, but they're being used in a different context. So the universality of biology, and also the importance of really understanding basic data. I think that's quite an important thing for people who are executives in biotechs, is to actually have a feel for what the data means, and what does a Western blot mean when you look at it. And unless you've spent hours slaving over data, you really don't understand, and you have to make quite profound strategic decisions on the basis of hard data. So I think that the years that I spent in the lab doing experiments and running a lab, I think were incredibly well spent. The other thing is that in the hospital, I was looking after patients, and also I ended up running the Use of Medicines Committee and looking at therapies from a payer's perspective. And UCL is a big group of hospitals, and we had about £100 million worth of drug spend a year. And So, you got a very clear idea for what the expensive drugs that don't work are, and the areas where the market will support expensive therapies. And I think that payer's perspective was actually incredibly useful. And to have that at the same time as looking after patients it was pretty unmatched in terms of an apprenticeship for going into the industry.
Very much so. If, as an aside, I might note that about 15 years ago, I was in a workshop where your father was present, and I remember him distinctly saying, "We ignore payers at our peril." Of course, the current situation has really confirmed that insight so long ago, when this was really not that common, to keep the payers
Absolutely.
in mind during drug development. So he was right.
No, I mean, I think that's absolutely true. And the interesting thing as well now with the big changes in the US, the question of value for money for the payer has really never come into it before, and that was always anomalous. And I think now it's absolutely logical that you have to get good value for money for these incredibly large spends.
Because you spent so many years in medical research, I want to ask you: have you come across any important differences between clinical trials in Europe and clinical trials in the US or other regions?
Yeah, I mean, the nice thing is that intellectually there aren't. It's the same framework, the same regulatory framework. And I think that's incredibly important, because in the end, data is data and patients are patients. But from the point of view of actually conducting the trials, I think in Europe at the moment, and I include UK in Europe, which is becoming a more fashionable view. It went out of fashion for a few years, but anyway, that's a separate podcast, I think. Europe is actually a very good place to deploy capital for clinical trials. There's a lot less competition for patients, so it's easier to recruit. The quality of centers in the main European countries is really excellent. And the speed at which you can do a clinical trial is probably higher than in the US. The US, of course, is going through some changes at the moment at the FDA level that will resolve. And I know that some US biotechs are actually looking at Europe as an alternative place to do clinical trials. So I think this could be Europe's moment. And I think the other anomaly in our situation is China, who had gone, had huge efforts to orientate their regulatory process with the US and Europe, and then because of politics and international competition, that sort of opportunity has sort of slipped. So I think at some point that will normalize, and so that will then become an opportunity for small companies like ours. But in the end, of course, we'll certainly be doing clinical trials in the US, and the quality of clinical trials in America is as good as anywhere in the world, of course. But again, the problem is the expense. Things tend to be a lot more expensive in the US, and so you have to be better capitalized. So at our small stage on the feeding chain, I think I'm pretty happy that we're deploying capital in Europe.
I've just come across the expression brain gain, referring to the opportunity that regions outside the United States have to, if you will, attract scientists and physicians, clinicians, researchers from the US to other parts of the world. Any thoughts about that? Is it a brain gain potentially, as opposed to a brain drain, which it had been in decades past?
Yeah, I know, it's an interesting one, isn't it? The big changes that have happened are the reverse diaspora, for example, of the Chinese researchers who are now going back to make China into one of the big drivers of pharmaceutical innovation in the world. So I think it's a really interesting question, the sort of movement of researchers transnationally. I think it really is one of those things. But you do make a very good point that people are mobile, and they're mobile between countries, and of course they're mobile between companies. So if you're running a biotech, you have to be aware that you are competing for your own staff every day. And if you are not the place that they want to be, they'll go off and work for your opposition. And there's nothing you can do about it apart from being the place that people want to work. And I think that's great. That's the way that capitalism should work.
So after a couple of decades in academia, you moved to industry. You became CMO and CEO at Silver Creek Pharmaceuticals, working on regenerative therapies. So can you tell me a bit about that experience and the impact of that experience on your journey?
I'm profoundly grateful to have been given the opportunity to do this, and I'm very happy that I took the leap. So moved to San Francisco and took up reins of this company, trying to do something entirely novel. The cultural differences between the US and the UK are actually quite profound in terms of work. Because we're immersed in US culture, we expect everything to be the same, but quite often it isn't. And it's, certainly the diplomats in the UK have been, they always tell you that when you go to the US, you really have to be aware that you're going to a very foreign country. Not necessarily, not in a bad way, but it's just different. And because we're lulled into this false sense of, we're the Americans and the Americans are us, we're always kind of surprised when they behave that we're not. And certainly joining an American company and working with an American team as a Brit, it was really interesting to probe the differences and to introduce some advanced British management tools to the Americans, like going to the pub after work to continue your discussion, which is something that they hadn't really done before, which is the way that UK medicine works. So that was great. The Californian ecosystem was eye-opening, to say the least. The speed and the aggression in a good way, and the amount of money in the system, really very, very eye-opening, and very grateful to have been exposed to that.
It's been said that America and Britain are separated by common language, and some of the examples you gave reflect that to some extent.
Completely right, completely.
Curious about how you navigated the transition from a long time in academic culture to the industry culture.
I have to say I'd been getting very frustrated at the time with the academic culture. The sort of, what I would call stamp collecting mentality of a lot of the people I was working with, just trying to publish the next little paper and get the next little grant. The thing that I really loved about the US in general, and Silver Creek in particular, was the ambition to do something entirely novel, and just to really go for it. So that actually was a very profound change that I went into with eyes open, and that's one of the reasons why I did that. In the end, I have to say, in general, the attitude of industry to data is far more honest and far more diligent than academia. The problem that academia has with reproducibility of data, it's very well documented, whereas the standards of data acquisition and data faithfulness in industry is an order of magnitude better. So I think that environment I've really I really have felt very positive about.
Is that difference the result of regulation of industry versus much less regulation of academic research?
I think that's probably true. But the other thing is, Henry Kissinger has a great quote. The reason that arguments in academia are so vicious is the stakes are so low. And so people would, throw stuff at each other and be consumed by petty jealousies and not give each other grants when they're reviewing them, and that's a huge problem. Whereas in the more cut and thrust environment of biotech, there isn't that level of pettiness.
Moving to the company you now lead, Arjuna, I'd like to know a couple of things. What does the name Arjuna mean?
Yeah, so our molecules are made from silver atoms and other atoms, but at the moment our molecules are made from silver atoms. And Arjuna was a Hindu god who killed evil things with silver arrows that never missed. So we thought this was an appropriate name for the company.
To dig a little deeper then into Arjuna, what inspired the formation of the company, and what made you believe that you could stand out as Arjuna in this crowded landscape of oncology?
So I joined a CEO group. I'm fundamentally a pharmacologist, and everything is to do with dose. And the dose of being English that is optimal isn't 100%. Likewise, the dose of Californianness isn't 100%. So I was trying to become more Californian and less English by joining a CEO group and meeting CEOs and emoting and doing all the things that the British people find difficult. Incredibly.
How did that work?
I think a massively positive experience, amazingly positive. One of the other people in the group, the chap who runs a very successful company making pigment, his side hustle was being the US head of a Spanish chemical spin-out. And he told me about these chemicals that they were developing, and they were thinking that they were going to be a drug at some point. So he asked me on behalf of the Spanish team what experiments I would do. And I sort of mentioned a few animal experiments. And 6 months later, I got an email from them saying, well, we've done the experiments. Here are the results. And they were amazing. And this was at a point when we were coming back, the family was coming back to Europe. And the Spaniards had heard about this somehow. And they said, well, why don't you found the Biotech? So that's how Arjuna was born, and the idea of these molecules is that they're going to be able to specifically inactivate the mitochondria of cancer cells, which is something that no other molecule can do. So this is potentially a game changer in the treatment of cancer, and you mentioned the crowded field in cancer, and you're absolutely right. It is crowded, but it's crowded in the same way that a football pitch with 5-year-olds playing football on is crowded. So there are crowds of people, but they're all following the ball. They're all following where the ball has been, and there are vast spaces where there is nothing. And that's sort of what people are discovering now in the biopharma world, is that everybody's put money into the same assets. I think the herding instinct in biopharma has been and is a huge problem.
Arjuna's approach is very unique when it comes to cancer treatment. Can you get into the role of therapeutic molecular clusters and how you are approaching clinical development based on that platform?
Yeah, so metastatic cancer remains the biggest problem in oncology, and really, unless you're very lucky and in a certain subtype, if you have metastatic lung, bowel, pancreatic cancer, your prognosis is pretty similar to what it was a couple of decades ago or more. It hasn't really changed. And it's become very obvious to the field, and certainly this is very obvious to us starting the company, was that the cancer isn't just a genetic disease. It's genetics, it's immunology, and it's also metabolism. So it's a stool with three legs. And it's become very obvious that simply giving genetically targeted drugs, but they're very easy to validate in animal models. So they've had a lot of uptake from VCs. But once you get them into patients, they don't make people live longer. So our molecule, our first molecule, AG5, is different in that it's a catalyst. So we think that AG5 eventually will be the drug that stops cancer metastasizing. And that's a big deal, of course, because it's the metastasis that kills you.
And at what stage of development is Arjuna now with your lead compound?
So we are preclinical. We are now starting GMP scale-up manufacture and starting our IND enabling work. So we're aiming in 2027 to do our first clinical trial. And our first clinical indication is using AG5 as a monotherapy in glioblastoma. So glioblastoma is at least 50% of glioblastomas are very sensitive to AG5, and we have a robust biomarker that will predict that. So our strategy is that we will show clinical efficacy as a single agent before going into combinatorial regimes that will then allow us to leverage the really enormous markets of metastatic solid cancer across cancer types, and then finally into radiotherapy, which is tens of millions of patients and no drugs. And we think that's going to be a profound change. for patients not only in Europe and America, but also in the middle income parts of the world where the majority of patients live, where the majority of cancer therapy is delivered by external beam radiotherapy.
Turning a bit more personal, there's often a personal why behind the work that happens in biotech all over the world. So I want to ask you, what is your personal why, and how do you stay connected to the human impact of your science as you're involved in this work?
When you're a hospital doctor, as you get older, you accumulate experiences where you've had to tell patients and their family that, I'm sorry, there isn't a drug that will help you. And telling a family that their mum is going to die and you don't have the tools to do anything about it is a very, very big deal. And I, you only, when you get to my age, in general, you don't tend to remember the patients who you've helped in general, but you remember these patients where you haven't been able to help them, and it does make you kind of angry, actually. And the other thing, of course, is as you get older, you become a patient, you become a parent of a patient, and you become a child of a patient as your parents get older, and certainly the experience that my father had. My father only got ill once, but that was pancreatic cancer. And so he had a pretty miserable last 6 months battling with metastatic pancreatic cancer. And he'd spent 40 years plus working in a hospital and loving it. We always used to think actually that he liked work more than his family, which is probably true, but hey, not uncommon, I guess.
Not uncommon.
But the big insight that he had at the end was that as a patient, he really hated being in hospital. So what he wanted from a drug was to stay away from hospital. And as a doctor, you blithely write a request for a CT scan, and then you see the data a few days later. But what you don't see in the middle is having to find a parking place, sitting for an hour or two, waiting for your scan in a cold corridor, going home exhausted afterwards. And all of that stuff he thought was terrible. So he went through all this just at the time that I was founding Arjuna. And so one of our key goals is to make a drug that is deliverable at home and keeps people away from hospitals. And that's really obviously extends their life. That's the other thing. People want to live longer. They don't want their progression-free survival to be longer. They want they want to live longer, and they want high quality of life, and they want to stay away from hospital. So I think of course, all of these things that I kind of knew, but until you go through it and it becomes emotionally stamped on you, you kind of don't properly know.
So that's what drives you at this time.
Yeah.
No doubt you've had to translate complicated science into a compelling story for investors, because money is needed for biotech to go forward. So what has been your experience raising capital at an early stage, and what did you learn in the process?
Nothing really prepares you for pitching to investors. And there are lots of different strata of investors, of course. And it became very obvious early on that I didn't understand the sort of underlying motivation of a lot of the people that I was speaking to. So I've spent a bit of time educating myself on this. And I think that certainly a lot of the investors who invested in us are really interested in the story, interested in the potential, but also are personally committed to the team, the individuals, and they're sort of investing in the people as well. And I think that's a really important thing. The interesting thing comes when you have the professional investors and the VCs and funds, and they are a lot more risk averse, of course, and so they want a lot more evidence. And we're certainly doing a lot of work now specifically for certain individual investment funds. But also, it's become obvious that VC as an investment class, in a lot of cases, and I don't want to tar everybody with the same brush, essentially isn't aligned with drug development in a very helpful way. They will make bets that are thinking about how they're going to get money into their fund rather than how a molecule is going to get into the clinic. There's lots of people saying the VC model is broken, but there's a huge amount of money going into it, and VCs have an enormous amount of power over a lot of work that's being done. But as I've said, there is clustering. A lot of VCs don't like doing something new. They want to invest in the same thing as everybody else because it feels less risky. Whereas, of course, that the opposite is true, because even if the science works, if you're the 15th company doing something, there is zero chance you're going to make a commercial win from it. So it's a really, really interesting point.
Powerful insight right there. Moving to clinical development, we all know that clinical development of a new drug, a new therapeutic, is always a roller coaster. So what have been your highest highs and most difficult lows so far in your career journey as a drug developer?
Yeah, I think, I have to say that I get high when anything works, because in research, especially in clinical research, it's so difficult to get anything to work. When anything works, it is absolutely wonderful. And as you get older, you mentally you sort of lower the peaks and raise the troughs. Otherwise, you would go mad. Nothing is ever as bad as it looks or as good as it seems. So it's the way to survive this crazy environment. But at the same time, the highs and the lows are what gets you addicted, and one without the other is meaningless.
You mean you need lows? You need lows. You don't just go for the high. You need some lows to explain that.
Yeah.
Get into that a little bit more.
Well, I think if everything you did worked, it would just become rather boring, And I think you need things that don't work, because if you've designed trials and experiments properly, you learn as much from things that don't work as things that do. And I think that's really important. And in the end, it comes down to designing your clinical studies in a way that gives you a genuine answer, a true negative or a true positive. If you haven't got adequate statistical power and you get a false positive or a false negative, then the next trial becomes difficult. And it's the classic phase 2 to phase 3 trap. The amount of money that's been put into phase 3s where it's failed, and then the analysis of the phase 2 that allowed it to go ahead has been found, well, maybe it wasn't as good as it could have been. So I think, by the time we get to phase 2, it's very easy for me to say now, but one would hope that we would be well capitalized enough that we would do a trial that would give us a proper answer one way or the other. And the other false negative, if you like, is the way that people behave. And so that, if you're talking about disappointments, the major disappointments that I've had haven't been to do with biology, because biology is biology and clinical trials are clinical trials. But the thing that you can alter is the way that people react when things go wrong. And this fundamentally comes down to who are the people you want to surround yourself with when you found a company, because these are the guys who are going to be in the foxhole with you. And I think that's the real thought experiment. Who do I want around me when things go wrong? You need people around you who aren't going to panic, because as you very well know, Alberto, there's 100% chance of things not working when you're doing biotech at some point. And
well, maybe 98%. Come on, we got to have a 2% of optimism in there somewhere.
At some point, not everything works all the time. And what you want is to be in a position where this doesn't destabilize things.
Yeah, everybody makes mistakes, as we know, and that includes drug developers. In your opinion and experience, what are some big mistakes that drug developers make?
I think doing the same thing as everybody else. I think fundamentally, the idea of we can try something that's 5% better than the incumbent therapy. I've always thought that was a mistake, because in the end, by the time that your drug is getting into the clinic, you're actually competing with the generic version of the current incumbent drug. And I think that commercially has been disastrous for a lot of cases. The clustering of money in companies around individual themes, I mean, is difficult because everybody thinks they're doing something that's differentiated, whereas actually they're not. And the other thing is thinking about clinical trial progression and thinking about learning about the molecule rather than trying to generate endpoints that will allow you to raise money. And I think that's a trap. I mean, it's very easy for me to say because, I'm not trying to raise money on the basis of a clinical trial. But if you're trying to be over ambitious in terms of the endpoints with the resources that you have, I think it's a recipe for disaster. And certainly a lot of really good assets have ended up just being destroyed by overambitious clinical programs that haven't tried to incrementally build value.
What are some blind spots that could cause the drug development train to, shall we say, go off the rails?
CMC.
Meaning I'd say chemistry, manufacturing, controls.
Yeah, manufacturing. Yeah? If you're in academia, you phone up Sigma and get them to send you 20 milligrams of the compound you want to test. And if you're in the clinic, in the hospital, you go to pharmacy. Whereas in our world, not only do you have to do the biology, you have to do the manufacturing. And for people like me, who's born and brought up as a biologist, suddenly I'm expected to think about process chemistry, and that's the easiest way for a small company to be torpedoed early on, is by having CMC disasters. So I think that's certainly a lesson that I've learned along the way, and it's difficult because for us, we're the first group to have ever made this class of chemical. So we've gone through the hard yards of the last 5 years of going from batches that don't work and we don't know why, to now we've got a really good understanding and we're able to go into a GMP scale up process with a really excellent manufacturing partner in Benelux. So that has been a journey, but the experience that I've had will certainly make me always think about CMC first before everything else. And I think that I think is the on my risk register is always going to be my highest.
That would, I think, apply to cell therapy and gene therapy, where you're manufacturing not in the classic sense of a small molecule in a large factory, but really almost at the bedside.
I think probably even more so.
Ross, you've worked both in the science-driven environment and business-driven environments. How do you approach, and have you approached your leadership role in a way that keeps your academic teams or industry teams focused, inspired, and resilient in the face of the occasional or frequent adversity?
Yeah, it's an interesting thing to reflect on. I think in the end, what you're doing is competing for people with your competitors, and I think that's the fundamental thing in the company sense. And likewise, in academia, people don't have to work for you, so you have to provide them with a vision of something that's going to be worth their while, either academically or financially. And that's very important. So just as you have to show investors a clear path to the clinic, people working with you have to feel what they're doing is worthwhile. The other thing is, I think, how you treat people. It's one of these sort of truisms that you see on LinkedIn. People don't leave companies, they leave their boss. And I think that's an important thing. And as I said earlier on, reverse mentors, people who've been people that you've hated working for and very glad when you don't have to work for them again. You don't want to be that guy. And I think that's very important. And so to think about Arjuna, we've got a small team, and deliberately we have a low number of people, but they're all really, really high quality. And so people are empowered to have control of what they're doing, and they're not micromanaged, and that's really very important. But the thing that I found the most gratifying is the emergence of leaders from the younger cohort of employees. So from the lab, we now have a VP of lab biology who emerged as a natural leader, and we're able to empower her and give her more responsibility. And she's grasped that. Likewise, our head of operations she makes my job so much more pleasant by just taking on board a lot of the terrible stuff. But in return, we're paying for her to do an MBA, and at the same time, she's had 2 children. So this is a high performing person, and having this group of people together and enabling people to grow like this is, I think it's the way forward, because the other thing is that things need to be fun, right? So we have to enjoy what we do.
In closing then, Ross, and looking ahead, what impact do you hope to have on cancer care in the next 5 to 10 years?
Well, I hope you've got the feeling that we're quite ambitious. So we're aiming that AG5 is standard of care for primary brain cancers in adults and children, and I think we're increasingly confident in the story. And this is one of the biggest unmet needs. But we want to go beyond that, as I've said, and our molecule is really the only way that we're going to provide the third leg of the stool of genetics, immunology, and metabolism to tackle metastatic cancer. And so our vision there is that our drug could be the drug that prevents metastasis, and part of the next generation of combinatorial regimes that turn cancer into a chronic disease, much the same way that HIV drugs have turned HIV into a chronic disease. That's what we're aiming for in the medium term. And the other really important area is external beam radiotherapy, because that, as I said, that's tens of millions of people worldwide with no drugs, and it has been a blind spot of big pharma. So I think that certainly the preclinical work that we're doing and the collaborations that we're developing make it increasingly likely that this is going to be a drug that will increase the safety and efficacy of external beam radiotherapy, and that hopefully will allow us to build a footprint in parts of the world like China. And certainly, I am a Sinophile. I mean, literally, my wife's family is of Chinese extraction, and we want to be doing clinical trials in China, but at the moment we're too small as a company, and we need to find a partner, and we need to find the right clinical indications. And I think external beam radiotherapy and also lung cancer are probably going to be that.
Well, I hope it works for the sake of patients everywhere, Ross. And that brings us to the close of our conversation. And that's it for this episode of Clinical Trialblazers. Thank you, Ross Breckenridge, for sharing your insights and experiences with us. I loved so many things about what you said. And in particular, who do you want to have in the foxhole with you for when things go wrong? It's such a profound insight on how important it is to select the right team members that will have your back and you will have their back when things go sideways. I really appreciate that and everything else you said. For our audience, stay tuned for more episodes of Clinical Trialblazers, where we will continue to explore human stories behind the ideas and innovations shaping biopharma and biotech. If you enjoyed this episode, check out our other episodes and be sure to follow or subscribe. Please take a moment to rate and review this podcast wherever you listen. Clinical Trialblazers is created by Biotech and hosted by me, Alberto Grignolo. Special thanks to our production partner, Wonder Media Network. Until next time, stay curious and let's keep pushing the boundaries of clinical research.